Good. So, very good afternoon, everyone. It gives me an enormous pleasure introducing today's speakers to you. The first one is Alessandro Tondelli from CREA Fiorenzuola in Italy. Alessandro and I go back a very long way, so we did a PhD together. And then I left the country. Probably this is a good thing for Italy. While he stayed and rose through the ranks of CREA, where today he's one of the leading scientists at the Research Institute. Together with Alessandro today, Alice Branchi is with us. Alice represents a new generation of Italian scientists. So she's a PhD student at the University of Catania. Is working at the same place as Alessandro as a barley geneticist. So without further ado, I would leave Alessandro and Alice the floor, and I look forward to hear your discoveries.
Thanks, Davide. Thanks for the nice introduction. So just let me share the screen. So hopefully you see my screen.
Looks good. Yeah.
Good. Okay. You can go so laser. Okay. So thanks, David, again for the introduction. Also, thanks to the organizer for inviting me to give this seminar. So I'm going to talk about what I think is a very fascinating topic, that is barley response to abiotic stresses. Also, is a topic that obviously is getting more and more important, interesting in the context of climate changes. And I'm just giving you here. So it's not moving here. Well, just to be very pragmatic, the impact of climate change at our site. So here we are located in the north of Italy. So in the last 25 years, we have already experienced an increase in the temperature, the average year temperature of 2 degrees. And this is mainly due to milder winter that we are encountering, you can see from this graph. So which is also the impact on our favorite crop, that is barley. So in this graph, I'm showing the results of nationalist trials. Actually, here is a single variety, the winter barley, 6-row feeding barley that names Ketos, a variety from France. And you can see here that in these 15 years, this variety has lost almost 20 days of the cycle. And also there is a slight decrease in grain yield. So this is something that obviously we have to face, and we have also to react to this trend by breeding. Also, from this graph here, you can see that the occurrence of very low temperature days are decreasing year by year. So many farmers also in Italy that are starting thinking about sowing spring barley in the autumn, like it's done already in Italy in the south, in the central part of the country. So this may be interesting also for malting barley, obviously. But there may be still a risk of low temperature that can damage the crop. So some years ago already, we started wondering if there is variation for low temperature tolerance in spring barley. It was already demonstrated in this very old work. So we started by looking at a panel of around 200 spring two-row cultivars from across Europe. And we were using a very high throughput screening for testing the damage of frost on the functionality of the photosystem tooth. So this was developed by Fiorenzuola in our lab. And then we were mapping genes related to this phenotypic variation that we have observed in spring barley. We were a bit surprised to find the same major QTS that also control the tolerance to low temperature in winter barley, that has frost 2 and frost 1. So just quickly to remind you what are frost 1 and frost 2. So they were firstly detected in the winter by spring barley cross Nord by Tremois by Enrico Francia 20 years ago. So frost 1 actually is corresponding to with the major venerization gene, 1, while frost 2 here is mapping in coincidence with a cluster of CBF genes. So CBF genes, they are transcription factors that are very well known in different species because they participate in the early phase of the plant response to low temperature, and they actually their main role is in the activation of what are called the code-regulated genes like Grf4 is reported here. So when we're studying then the locus at the genomic level, we observed that actually in winter barley Nure, there was a tandem duplication of a genomic fragment of 22 kb where CBF2 and CBR4 are located. So probably this is the copy number variation, and because of this copy number variation, CBF gene are higher expression in Nure with respect to Tremois under low temperature. And as a consequence, they have a higher induction also of code-regulated genes that are the genes that actually then are responsible of making the plant more polar. So more recently, Davide Guerra in our lab, he was interested in widening a bit the diversity and looking at differences in the diversity in barley for these two loci, for first one and first two. So he decided to screen the Wilby collection for the lead mining for these two loci. So this Wilby collection has been already mentioned a couple of times in this series of seminars. Probably it was worth also spending a few seconds describing the collection. I think it's very nice representation of the barley diversity. It was assembled by Benjamin Killian when he was in the HiPK during the Wilby project. It was coordinated by Robbie and Gilles Charmet. So in the Wilby collection, we have around 240 old and modern cultivars. Then we have 240 landraces from all over the world. You can see here in this picture. And we have also some wild barley, including the parents of the HEP25 population from Glaspillen. Then what is really relevant with this Wilby collection, I think, is that there is very good genomic data. So the coding portion of the genome has been captured and sequenced. And the sequencing was actually done very high depth. So I think that we have very robust SNPs, but also structural variants. And more recently, we have mapped also k-mer related to this data. And actually, because of this very high sequencing depth, Davide was trying to look at copy number variation of CBF genes in the Wilby population, just exploiting reads count. So he was mapping the reads at the CBF2 and CBF4 gene in the different Wilby sessions. And then he discovered that actually there is a bunch of lines that has a very high reads mapping at these two genes. And when he was normalizing this read count for the length of the gene and considering a gene that is present in a single copy in the barley genome, like this SMDC, he could observe that actually there are accessions having a higher numbers of CBF2 and a higher number of CBF4. And actually these numbers are clearly highly correlated. And this is actually confirming that CBF2 and CBF4 probably they lie on a genomic region that is then randomly duplicated in some accessions. Those data were validated here by digital PCR. You see that digital PCR data really confirm what we have observed from all the exome sequencing. And then what is really important in this graph is that you can see that most of the accessions here, in particular, sorry, spring barley, are located here. So they have just one copy of CBF2 and CBF4. But here you can see that winter barley, they have larger, more copies of CBF2 and CBF4 than facultative barley. Usually they have even more copies of the two genes. But what is really interesting that the accession with the highest number of CBF2 and CBF4 are 4 spring barley, 6 row from Nordic countries, from Russia, from Norway, from Sweden. So obviously we were interested to see if these 4 accessions also have a higher temperature tolerance to low temperatures. So in this case, the frost tolerance screening or test was done in the lab of Gabor Galiba in Martonvaja, and I can say that we were a bit disappointed to see that there is no clear correlation between the copy numbers of CBF4 in this case and frost tolerance. In fact, we see these two lines that have high frost tolerance independent on the number of copies of CBF, and the same for line with low tolerance to low temperatures. On the other side, there is a clear relation between frost tolerance and the expression level of the CBF4, in this case under low temperature. So still, we are wondering why these two accession, for example, they have such a large increase in the genomic fragment passing CBF2 and CBF4. So maybe, well, sometimes this might also come to a cost for the plant. So for example, we know that plants overexpressing CBF genes, they have a stunted growth. So we really have to understand why these genotypes have a large number of CBF genes. Maybe that we simply are not hitting the right timing, or maybe these CBF genes are related also to other abiotic stresses. Then obviously, Davide was interested, other than copy number variation of CBF2, also in variation in other CBF genes. And in this case, he found a significant association between frost tolerance and a specific haplotype at the CBF14 gene that was already associated to frost tolerance in barley, a paper from Agostino Fricano, and also from a paper in Martin Paglia. And recently, I was reading this nice review from Eric Stockinger. He's the father of CBF genes, and he actually was claiming that there may be more functional alleles, FOS2. So the neural allele, that is the one with number variation of CBF2 and 4, seems to be present in many winter barley varieties, but also there may be another functional allele that has been detected, for example, in this Korean landraces, that has some similarity with the spring alleles, and also it seems it have a higher copy number of CBF14. So it's something that we are trying to investigate right now. So I just gave you here the major results from this paper that has been published a couple of years ago. Obviously, if you are more interested in more detail, please go and read the paper. But I want to switch now to another very nice experiment on the Wilby collection. And this experiment was led by Eva Maria Gomez Alvarez and Chiara Pucciariello from Scuola Sant'Anna in Pisa. So they are in the lab of Pier Domenico Barata, where they have a very strong track record of studying anoxia and hypoxia also in crops, mainly in rice. So Eva was interested in study variation in the Wilby collection for the ability of barley to germinate after a short period, 2 days of submergence, followed by 6 days of recovery in hair. And actually, she was able to find a very large variation for this trait. So here is the percentage of seeds that germinate in this condition with respect to hair. And here, there are a picture that is showing two very contrasting genotypes from the Wilby collection. So when we were running this data in GWAS, we actually detected a major locus on the chromosome pH, and the best candidate for this locus is actually lacase gene that is ortholog to Arabidopsis lacase 5. This gene in Arabidopsis is controlling the accumulation of lignin in the seed coat and, as a consequence, probably seed permeability. So in this paper, they were using this test, tetrazolium test, showing that mutant in LAC5 in Arabidopsis actually have a higher permeability. And Eva, running the same test, she was also able to demonstrate that tolerant barley lines have a higher permeability with respect to sensitive lines. So at the molecular level, this is the gene. So we have a few SNPs in the coding portion of the gene region, but we have also an indel in the promoter that distinguishes between the sensitive and the tolerant genotypes. And then Eva was looking at how this gene is expressed during the development of the spike and the development of the grain. And she observed that there is actually a higher expression in sensitive barley during the tough stage in seed ripening. So probably this higher expression is leading to a higher accumulation of lignin in the seed coat, and as a consequence, sensitive barley, they have a lower permeability. In fact, she also was observing that sensitive adjacent here have a lower absorption of oxygen in the submergence water with respect to tolerant line, and this might lead to a very rapid state of hypoxia in these barley seeds. And actually, to confirm that there is a state of hypoxia, Eva was studying the expression of the alcohol dehydrogenase. Is a well-known marker for fermentative metabolism. And you can see here that the sensitive line has a higher expression of alcohol dehydrogenase with respect to tolerant lines. Then hypoxia, it's well known that is also inducing what is called a secondary dormancy in seed. And this is actually due to a higher activation and sensitivity to ABA. In fact, Eva was observing higher content of ABA in sensitive seeds with respect to tolerant seeds. And also she was observing a higher expression of another well-known marker, AB5. In this case, this is a marker for seed dormancy. So sensitive line have higher expression in this AB5. So probably they are entered in this secondary dormancy state. And this AB5 actually is the activity of this AB5 is reduced by nitric oxide, and for this reason, when we treat the sensitive line with these molecules that are donors of nitric oxide, the activity of AB5 is decreasing, and then these lines are able to germinate. And on the contrary, if we treat tolerant line with another chemical that is actually a nitric oxide scavenger, these lines are not able to germinate because they enter the secondary dormancy. So this is the model that Eva was developing actually during seed development in sensitive barley lines. There is a higher expression of lactase, so there is a higher accumulation of lignin in the seed coat, and this under submergence brings to a lower permeability of the seed, so oxygen cannot enter the seeds actually, and the seeds will enter in a hypoxic state and they will activate an IBA network that leads to a secondary dormancy. And to finally prove this model, Eva was working with a interesting method. So she was mechanically removing the seed coat with sandpaper. So you can see that sensitive line here, when they were scratched, they were able to germinate even under submergence. Also in this case, I'm giving you just the major results. It's interesting. So if you want more details, you can read the paper that was published last year, Journal Experimental Botany. But now, finally, I want to move on the opposite side because the last couple of years, we are studying also the response of drought, and everything started with this experiment that was run by Menachem Moshe from the University of Jerusalem in the frame of the CleanBar project. So he was testing 81 spring barley under the plant array platform. Probably you know this platform. We have also bought 72 units in our greenhouse. So this is a gravimetric system where you can measure in continuous the amount of water that you give to a plant and the amount of water that is transpired by the plant. So in a classical experiment that is shown here, you have a first pre-treatment phase where you give an excess of water to the plant in the night, so that in the morning the pot is at full capacity. And you see that every morning when the pot is full capacity, you can observe an increase in the weight system. So this is an increase in the biomass, and you can relate this increase of the biomass to the amount of water that is transpired. So in this phase, you can estimate the water use efficiency in a precise way. Then you enter in a second step of the experiment when you stop watering. And in this case, you can see that different genotypes behave in a different way in terms of their response to water shortage. So there are lines that are called isohydric here, that they feel that the water is decreasing in the pot, in the system, so they tend to limit the transpiration. But on the other side, here on the opposite side, there are lines that are called anisohydric, so they continue transpiring even if the water in the pot is decreasing. And then there is also a phase of recovery. Actually, just a couple of days ago, paper came out in Frontiers of Plant Science describing this experiment. But what's really more interesting here for us is that based on this data, few accessions were selected because they were contrasting for their response to drought, and they were further studied in the plant array, in this case in a different experiment, in a different platform, in the plant array of Raimund Rotter at the University of Göttingen in Germany. And here we focus on two different genotypes that are Chanel and Formula because they have a contrasting response to the shortage. So you see here under control condition, the y-axis of this graph, there is the daily transpiration. So you see that in Chanel, the daily transpiration is always higher than in Formula, that is here in gray, under control condition. But then when you stress the plant, so drought condition, you see that Chanel, say, limits the transpiration in a much faster way with respect to Formula. And you can see also here graphically that there is a sort of penalty in transpiration that is much higher in Chanel with respect to Formula. And Mercy Appian was also calculating what is called the beta crit, that is the volumetric water content, which the plant starts feeling that the water is decreasing and limits the transpiration. So you see here that Chanel has a theta crit that is lower with respect to Formula, even if Chanel is transpiring at a higher rate. And interesting, this behavior that was observed in this case in controlled condition in a greenhouse under the planetary platform was also confirmed in a very small experiment run by Ernesto Garcia in Spain. So he was growing stem plants of Chanel and Formula under drip irrigation and under rainout shelter. And then he was measuring a few basic biomass or let's say yield data. And even if in few plants, so you can see that the trend is the same. So there is a larger potential, I would say, for Chanel. So under control condition, the biomass or the yield of Chanel is much higher with respect to Formula, but then there is a higher penalty in Chanel with respect to Formula. So we can say that Chanel is aligned with a high potential, probably a lower stability. Actually, Ernesto thinks that Formula is irrigation resistant. We can see from different views. But what is really important is that this is not driven by phenology. So Chanel and Formula more or less have the same phenology. They flower more or less at the same time. They have the same allele major flowering genes. And this, I think this is quite important because often or sometimes when you then want to map genes for abiotic stress, you end up with flowering time genes because you are mapping up with something that is not tolerance, but is more escaping the stress. So in this case, it looks like Chanel and Formula, they really have two different contrasting behavior in their response to drought. So we decided to study the genetic point of view, this differential response. And for this reason, together with Klaus Holtag, AWS, we have developed a double haploid population, and we have made a first preliminary test on the plant array platform in Jerusalem with Menachem Moshe. That time, unfortunately, there was room for just 2 pots per genotype. Considering this condition is well, semi-controlled condition, probably the design was not very robust, and the data that we got, they are not very robust and reliable. And also the heritability that we have calculated for different parameters, they are not very high. And as you can imagine, also the QTL analysis was not super, let's say. We were able to detect just a few QTLs for some traits like dry shoot weight or recovery rate. Here, this is the difference in transpiration between the pre-treatment and the drought phase, let's say. But the variance that is explained by this QTL is around 10%, so it's not zero, but they are not major things, I would say. So I'm not going to speculate about any possible candidate gene that are below this QTL, because we need to run another experiment, have better phenotyping. Also, we are trying to populate a bit more this data set with transcriptome data. So we were mimic an experiment like in the plant array and controlled condition in the growth chamber, growing Chanel and formula under different soil water content. And then we were sampling leaf tissues, different steps, these experiments, and sent out RNA for RNA-seq. And actually, the data were expected to arrive by mid-June so that they could present something to the seminar, but they arrived just this morning at 11. So I downloaded them, and then I will have something to do this summer. And also, we are trying to understand a bit more the difference between the parents, Chanel and formula, both at the physiological and also the morphological level. And for example, here there is a nice experiment that Davide Stan, so he has developed, has applied this method, where he uses a handy microscope to take high resolution pictures of leaves from different genotypes, and then he's using a machine learning model to recognize and count the stomata in the leaves. And based on a few data that are generated up to now, you can see that Chanel actually has a higher stomatal density with respect to formula. And this might be one of the reasons why Chanel is conspiring more than formula. Okay, so well, why I decided to present you three different abiotic stress instead of focusing on just a single one, and probably give you more details. Maybe I made some confusion. Actually, the point is that with good phenotyping assays, with molecular data that nowadays are available everywhere, and we can find for sure interesting resistant genotypes and genes in the wide valley diversity. But then we have to remember that the abiotic stress, they rarely come individually, but more often they occur in combination. So we are really to start moving a step forward and start thinking about multi-stress resistance. And also, you have to remember that another effect of climate change probably is this much greater here-to-here climate variability. As I mentioned before, starting the seminar, this year we have already 700 millimeter rain from January, but the last 2 years we have less than 200 millimeters. And on the graph here on the left, actually, this is from a paper by Gilles, where he was claiming that this year-to-year variability may be one of the causes of the yield stagnation that has been observed in wheat in France in the last years. So obviously, in this condition, it's not easy for a farmer to choose the good variety to grow, but also for breeders to choose what to breed for. So really, I think we have to move to multi-stress analysis and then experiments. And just to conclude, I see three possible options. So one option is that we can screen a collection for many different stressful conditions and then try to model the response, identify genes and alleles that may have a role in single or multiple stress. So, for example, this is an experiment that was run on Arabidopsis. They were screening a collection for 18 different biotic and abiotic stress, and then they were modeling multi-trait GVAs in order to find significant associations or genes that may be related to one or more stresses. And well, we were talking about football before, so I don't know if you like football. I hope yes. And then since it's very actual, I try to imagine these genes like a great football player with different role, different teams that now we have to put together. And after I was thinking that in this will be collection, we are starting accumulating quite a good massive amount of data related to abiotic stress, but not only. So I showed you about subenergence tolerance, something about post tolerance, but also we have stomatal data. So David has analyzed all the collection for stomatal density. Then we have root rates, both in our lab with Agostino, but also Silvio Salvi in Bologna. He was analyzing for root rates. Then we have some pathogen resistant data. So it might be worth seeing if such an approach works also in our collection. Then we have a second option I found here that is obviously to make experiments with combined stresses. So, for example, in this experiment from Fiorenzuola in Australia, they were growing a collection of wheat in a field where both drought and heat was experienced. And in this case, we can find what are called QPLA for the multiple resistance and like normal that they can do what they want. So this is another possibility. Then there may be a third option that is claimed by someone that is growing a varietal mixture because they, according to some researchers, might increase the yield stability. And this is an example that was done on winter barley. I think that we need to put some more science on this topic because very often it's just empirical. We need to understand which are the synergies between the varieties that are mixture or varieties and micro, for example, that can really provide a higher yield stability to these materials. And obviously, this is a teamwork. So here we are. Yeah, I will leave this for the discussion. So I stop here. And I just want to thank people that was involved in this work. So mainly Davide, who are in our lab, also Istvan in Hungary, then Eva in Pisa, Ahan Menakim in Jerusalem, Ernesto in Spain, and funds in our lab these days are provided by two European projects. One is SCROP, that is an Horizon Europe project fund, coordinated by Paolo Pesaresi, and the other one is Recoba, that is a FACE project coordinated by Ernesto in Spain. So I thank you for the attention, and I stop sharing my screen. And David, if you don't mind, I can introduce Alice.
Please go ahead.
Okay, so Alice Branchi, she's a second year PhD student at the University of Catania, and she's actually in Catania, so she has classes over there. But she is performing her experiments in her research in Fiorenzuola, in our lab with Cristina Corsati and with Raffaella Battaglia. So when I asked, following Robby's suggestion, I asked her to speak at this seminar because it's a good opportunity also to introduce new early career scientists. So she was very happy. That's why she's here. And so Alice, the floor is yours.
Thank you, Alessandro, for your introduction. So I'm going to share my screen. Okay, can you see the presentation?
Yes,
looks good.
Okay, so hello everyone. I'm talking about my PhD project, and my PhD project aims to study a gene, which is a Grf4 gene in barley, that seems to be a gene controlling some components of grain yield. So we're talking about potential yield, which is the maximal yield that a crop can produce per land unit when the conditions are not limited in terms of water and nutrients, and where there are no pests, diseases on the field. Yield is a complex trait that can be divided into some subcomponents, such as, of course, grain yield components, which can be divided at the same time into subcomponents. And these are, for example, spike branching, tillers, number of spikes, grain number, and grain size. As it is already known by the literature, grain size and grain number are in a sort of balance. So when trying to modify the size of the grains, usually this can produce a negative effects on grain number and vice versa. But at the same time, it is crucial to know the genes that can in some way affect grain and potential yield in order to study how to adapt those genes and to have positive effects also not only on potential yield, but also on yield stability in terms of, for example, of more stability on these weather conditions that are changing because of climate change. So the gene I'm working with is a growth regulating factor. These are transcriptional factors which are plant specific. Just to give you a brief idea of how these proteins are structured, there are two conserved domains at the C terminals of the protein, which are the WRC domain, which is the DNA binding domain, and the QLQ, which is a protein-protein interacting domain. So GRF's gene interacts with GIF, which stands for GRF interacting factors, and these two proteins targets the promoter region of their target genes and recruits the transcriptional machinery. GRF genes are regulated at the post-transcriptional level by microRNA 396, which controls their abundance. And the module GRF, GIF, and microRNA 396 is highly conserved among plants and can be a suitable module to be studied by functional genetics, since it can be modified at different points, for example, by modifying the promoter of GRF genes, but also by modifying the microRNA 396 target region on the mRNA of GRFs in order to change its regulation by the microRNA. In particular, I'm studying GRF, which is the orthologs to Oryza sativa Grf4, and this gene is known to influence yield potential. This was proven in durum width, where on some recombinant lines between Triticum durum svevo and Waldemar zavitan, there was found, some alleles were found to be correlated with larger seeds and also with a higher 1,000 kernel weight. And of course, this gene was studied deeply in rice, where it was made clear that this gene has some effects on the grain length, has some effects also on panicle length, panicle branching, and also on cell division. But in rice, it was discovered that this Grf4 was negatively regulated by DELA proteins, and these DELA proteins stops the binding between Grf4 and GIF proteins. And as always in rice, Grf4 seems, when it is overexpressed, seems to be to have a positive effect on nitrogen use efficiency and also on carbon fixation. So all these important functions may have a positive contribution to yield. And for these reasons, these genes is a perfect candidate gene to be modified and to be studied in order to know these genes affecting the components. I'm working on barley. In particular, I'm working on the cultivar Golden Promise, which is the most suitable cultivar for transformations mediated by Agrobacterium tumefaciens because it has the higher regeneration efficiency. We discovered that in this cultivar there are 18 GRF genes, and we are now focusing on to studying the Grf4 plate, where we can observe these two genes, Grf4a and Grf4b, which are the orthologs of Grf4 of rice. And so it seems that in this golden promise cultivar, the GRF gene is duplicating. And also we are focusing on this other gene that we call Grf3d, which is the orthodox to Oryza sativa Grf3. By some preliminary transcriptomic studies performed on public databases such as BarleyXDB, the role and the expression level of GRFs belonging to the Grf4 clade was made clear. So it was evident that this gene is mainly expressed in young spikes and also in some florets components. And it seems like these two genes, Grf4a and 4b, may be redundant and may have in some way the same roles. So during my first year of PhD, I produced and started to characterize a panel of genetic material. We produced some barley lines and also Triticum durum lines, where we performed an ectopic expression of Grf4a, and we expected to find out in these lines the Grf4 expression is much higher if compared to the basal line, which is this red line here down in the slides. Also, we produced some lines where we ectopically expressed Grf4a, but we modified the CDS of the gene in order to abolish the recognition site of microRNA 396 without changing the amino acidic sequence. So we expect this line to have a much higher expression level of Grf4a because we uncouple these genes by the post-transcriptional regulation operated by microRNA 396. We have also some lines where we produce expression of microRNA 396. So here we expect to see a downregulation of Grf4 gene. We produce some genome editing lines using CRISPR-Cas9 constructs. And in these lines, we expect to knock out the Grf4 gene. So we expect not to see the protein in these lines. And also, we are studying and trying to characterize some finding lines produced by Carlsberg Research Group. These are a non-GM approach for screening variant population in order to find out SNPs and some modification in some specific alleles. And by now, we have different time points and different time points and different generation for this genetic material. The most advanced are the ectopic expression of Grf4a and the lines where we perform the ectopic expression of the modified Grf4a. And here we are respectively in T3 generation and T2 generation. So right now I'm showing you some of the results that we observed in these two lines. So at first, in plants in barley plants overexpressing Grf4a, we can observe some plant morphology differences. For example, the plants with the construct seems to be a little shorter, and the leaf angle is different. We can observe some differences also on the spike morphology. For example, it seems in some spikes, we can see that there are slightly longer and a bit thinner than the control. And we observed also, we also observed some at some level of sterility, in which is not fully penetrant. So these plants produce fewer seeds, statistically fewer seeds if compared to the control. But the seeds that these plants produce are longer than the control. Even if you are the barley geneticist, I would like to show you also our results that we observe on the Triticum durum line overexpressing microRNA 396 sensitive Grf4a. Here too, we can observe some differences in the morphology of the plants. So the control parents are much higher than the overexpressing plants. Here too, we can observe some differences at the level of the spikelet morphology. In fact, the overexpressing plant seems to have more flowerets on the spikelet if compared to the wild type. And this phenotype here was observed also on the T0 lines that we transformed this year with the constructs overexpressing the normal Grf4a. And this phenotype was also observed in Triticum aestivum by one of our international collaborators, so Professor Wisterfeld from the University of Haifa. And he overexpressed the normal Grf4a, the same that we used in barley and in this transformation in Triticum durum. And the phenotype he observed was really similar to the ones we observed too. And in this case too, we can see that the plants produce fewer seeds if compared to the control plants. And these fewer seeds also seems to be longer, but also shrunken if compared to the control and to the wild type. So summarizing our first preliminary results, we observed some differences in terms of plant height and leaf angles in barley, and also plant height in the width. We observed some differences in spike architecture and in spikelet architecture in the width. In barley, we observed a not fully penetrant phenotype concerning sterility. And in both species, so in barley and wheat, we observed that the seeds length is really differentiated. So the seeds of the transformed plants and the positive plants are longer compared to the control and to the wild type. But since our first preliminary results have to be integrated with the results we will observe in the genome editing lines, and of course also find it lines. And all these results have to be integrated with the phenotypic data and metabolomics data that I perform at IPK in Gattersleben with Dr. Borschuk and Dr. Volodymyr Radchuk. We would like to integrate them also with a much more detailed transcriptomic analysis performed by an RNA-seq on developing spikes and young seeds, so the tissues where we hypothesize that these GRS are much more expressed. And we would like to perform also DAP sequencing. So we would like to study the targets of our Grf4 gene. And this will be done in collaboration with Professor Andrea Gallavotti from Rutgers University. And once we know the pathway of the Grf4 gene, we could dissect more in depth its role. And also when we will know the targets of the Grf4 gene, we can understand which other genes can be modulated in order to obtain maybe a greater effect on the yield without having those pleiotropic negative effects we saw on our overexpressing plants. So I would like to thank you all for your attention and to thank my supervisor, Dr. Raffaella Battaglia, Cristina Crosatti, and Professor Erika Mika, and all the other people which are collaborating with me.
Okay, so thank you very much, Alessandro and Alice, for this very interesting discussion and showing us your results. So I'm sure there are questions, and I read already a few of them in the chat. So I suggest we start with those. If anyone else has a question, so raise your hand and ask directly the speaker. So what shall we do here, Robbie? Can you ask directly Alessandro your question? Shall I read the chat?
I can read it, or you can read it. It doesn't matter. Or Alessandro can read it.
Oh, I can read, yeah.
Okay, so anyone can read, please go
So I
ahead.
can read. So the first question, Robbie, you're asking if there is this basic expression of CBF2 and CBF4 and the number condition is correlated with copy number variation. Yeah, I mean, these genes are really induced by low temperatures. So the basal expression probably is, in our case, was not detectable. So it was showing this graph and there were no differences under control condition between any of the contrasts. So then the point is, when these CBF genes start to be induced by, well, the control is this ICE1. So at which temperature? So which is the basal temperature at which the plant starts sensing the lower temperature, and then you start activating the CBF2 and CBF4 or other CBF genes.
Yeah, I mean, I asked because, am I unmuted here? I asked because in some work we did recently, it wasn't our experimental work, but looking at transcriptomics between different barley genotypes that had different copy numbers of CBF genes, we did observe a correlation between gene expression levels and copy number variation, but we hadn't done the experiment under cold-induced conditions. And what we were wondering was whether or not there was a kind of priming of cold response in the genes that got high copy numbers, so that they were much more able to respond to cold temperatures. You know, if it was a fairly rapid thing, if you know what I mean.
Yeah. But if I can add, when I was working in Eric Stockinger lab in the US, he said that once he was simply moving plants with a box from one office to the other, and then this was enough for making the CBF gene to be induced. So it was really strange.
Old offices.
So even simple movements were enough to observe a change in expression of these genes.
Okay. Yeah. The second question is a dead simple one, Ali, and that is you talked about a lot of data that you generated on the Wheelby collection. I just wondered if you'd put that data into the database, which contains all of the other Wheelby data.
No, actually, I think you are the person that were providing most data on the Wheelby. So we were downloading the raw data from what we have put in this rack and CBI, and then we were calling this schema. For example, is the last work that was done by Davide, but then we well, still have to validate the schema, and probably we will put on any kind of repository. And also the phenotypic data, they are from different sources. So I really would be interested to try joining together, probably once they're published and made available with the different researchers. Be interesting. But then there will be portal. I mean, from the website from the project, probably is not working anymore. So
no,
but I mean, we can put everywhere.
No, I mean, that's important because the value of that resource is only reflected in the value of the data that people can access.
Then I read the third question. Davide or?
Yes, please.
Chiara, ciao Chiara. She was asking if the difference in the stomatic density that we have observed between Chanel and formula was under control condition on response to stress, and also if we saw differences in cuticle thickness, composition, or wax. So as for the stomatal density, actually these were under control condition, but the ontogeny of the stomata is defined quite early, and since our stress was done at the booting stage, so when the flag leaf already was almost completely developed, I would say, yeah, we couldn't see in that experiment differences between drought and control condition. For sure, Davide is testing if there is a priming effect of, in this case, of drought on the, for example, what is imposed at the early phases of development. If this stress in the early phases is changing or affecting the stomatal density in leaves that are developed later on. So it's something that Davide is testing at this moment. And as for the cuticle, we didn't observe any difference in cuticle. I mean, we were not looking at cuticle, something that we can try doing. The work. And as for the waxes, well, in the field, they are for sure, they are not glossy types. So they are a standard spring to raw malting barley. I don't remember any glossy phenotype among these two varieties.
Can I add something maybe? Hi, Alessandro. Yes, I guess it's not really between glossy and wax in this case, but there might be small differences that maybe they are not even appreciated by eye in cuticle. And I will look. I mean, there is often a link between stomata changes or stomata density and wax accumulation. So yeah, I will look at it as well.
Yeah.
Can I ask
for the suggestion?
Can I ask something about the CBFs as well, since I'm here? If you notice, or if I've looked, if there is a difference in, I think it's called like induction temperature. So, for example, if the CBFs in spring or winter genotypes, do they respond at different temperatures?
Other than different spring temperature between, for example, spring or winter genotype, I remember in wheat, they claimed that the first sensor of low temperature is CBF14, and then CBF14 is activating other CBF. So there may be probably a CBF that is sensing the temperature before the others, and then the signal is actually spread to other CBF and other what they call regulated genes. Yeah, this is the only thing that I remember. We did not observe. Yeah. I can understand. I mean,
if be a different thresholds in different genotypes, I assume.
Yeah, exactly. No, I don't remember experiment that we have done in this sense, but it's something that also might be very useful to worth having a look. Yeah, sure.
Okay, thanks.
Then the next question is for Alice. Sorry that I'm taking your role.
No, go ahead. I mean,
So Robbie asked me, for your ectopic expression experiment, what promoter did you use? Did you generate constitutive high expression? We used ubiquitin of maize as a promoter, and yes, we generated constitutive expression.
So did you notice any other phenotypes associated with these high expression lines? The ectopic expression lines?
Yeah, these ones were the most promising. The other one have still like more in detail studied, because for now we focused on these phenotypes there.
Is that Mitchell? Mitchell is singing. So, yeah, what were the other phenotypes, Alice? Alice, sorry.
Yeah. Sorry.
Did you have dwarf or tall plants? Did they have multiple tillers?
We observed some effects on the height of the plants more than on tillers by now.
Okay. Okay, but the effects were pleiotropic, basically. Interesting.
Thank you.
Yeah, if I may jump in before Luke here is a follow-up question for Alice. So there is a trade-off in plants between growth and defense. So if you allocate resources to growth, you allocate less to fend off potential pathogens. So I wondered whether you said that you haven't seen many obvious pleiotropic effects, but if you be looking at also potential immune compromission or something related to immune responses that may be affected by your construct as well, if this is something that you have planned.
Well, I mean, by now we are trying to understand when and whether these genes are expressed, because of course we cannot say that much by using some overexpressing plants, because the protein is everywhere and all at once. So we would like to have before the data coming from the genome editing plants and the find it lines, in order to see if if we can see some pleiotropic effects on these plants too, and to try to understand whether these pleiotropic effects are due to the fact that the protein level is too high on our plants, or is due to something else. I don't know.
There's another question, Alessandro, in the chat from Luke.
Yes.
Oh, yep.
Yes, thank you for both of you for your talks. Two nice, clear talks. Alessandro, it just struck me when you were talking about the work you did on the Wilby collection. Was there an obvious correlation of the germination after submergence with any ecogeographical origin data or molting use, or any thoughts on the selection of the trait?
Yep, this is really interesting. So in the first version of the manuscript, actually, we ran a sort of more than ecogeographical. We were looking at the rainfall pattern during the growth of the plants in different regions, and there was a correlation between the tolerance and the amount of rainfall that these plants were having during the year. And this probably was not really convincing for the reviewer. That's why we removed from the paper. but there were at least some basic indication toward this that makes sense.
Mm-hmm.
I mean, plants that are experience more rain during their cycle, probably that they're accumulating this kind of resistance. Yeah.
It's nice.
I had another one for you.
And then another one is like case expression during seed development, environmentally sensitive in either sensitive or resistant type. What do you mean for environmentally sensitive?
So, I mean, if you grew either the sensitive or the tolerant type under different conditions, so either wet or dry, and then did your experiment on germination, would you get the same results or different results?
I think that the lac case, it was simply the expression of the case was monitored during the development of the plant in standard condition. So the plants were not grown either in lots of water or dry condition, more dry condition. So it was standard growing condition, yeah.
And the experiment you did showed it affected, or you suggested it was inefficient oxygen diffusion. But could it not also be water uptake?
Well, in this case, they were measuring with the sensor the amount of oxygen that was in the water that was used for submergence. Okay, so obviously then maybe also that the plant were getting less water, but I don't know how much big can be the difference. But also in the supplementary material of this paper, there's another experiment where they were using a sensor, a pin going into the seed coat to measure the amount of oxygen along a gradient in the seed coat. And this was another supporting evidence for lower oxygen penetration into the seed.
Yeah,
so this was another important point.
I asked the question, and Chiara will know about this as well, I think, because we were contacted earlier on this year from a malting company, or a distilling company actually, who had identified that grain they got basically last year was completely different from the grain they got this year of the same cultivar. And that the difference seemed to be in water uptake or in husk thickness. And I just wondered whether or not there was some kind of interaction between the lactase and this phenotype that they observed in the malt house or the distillery.
Yeah,
interesting to try.
Yeah, sure. Thank you.
Very nice data then.
Oh, a good suggestion also, thank you.
So I don't see any other question in the chat. So yes, unless there is any other question from the people online, I think we can close here. And please join me to thank again Alessandro and Alice for their seminars. Well done.
Thank you.
And I think that the only thing that it remains is please stay tuned for the next appointment. No, there's something else, Robbie, please.
No. I was trying to put a clap on, but I just thought I'd leave.
Okay, so thanks again, everyone. Please stay tuned for the next IBH webinar appointment.
Thank you.
Bye.
Thank you. Bye.
Bye.
Bye. Bye.
Thanks, Alice. Thanks, Ale.
Ciao.
Ciao.
Goodbye. Thanks.